Second is Dr. Deepak Kumar. Dr. Deepak Kumar,
Yes, ma'am.
please start. You have 2 minutes. Please go ahead. Your time starts now.
Thank you.
Please skip the introduction part. Okay, just start
Hmm.
with your oil presentation. Dr. Deepak Kana.
Yes, I am.
Dr. Deepak, can you please start now?
Yes, ma'am. Yes, ma'am. Screens.
We don't have that much of time. In 1 hour, we have to listen 24 candidates. Next candidate, please. Our next candidate is Dr. Shubhashi Sahu. Is she present? Shankala Mishra. Dr. Sangeeta. Dr. Sangeet. Sati Kumari. Dr. Deepa J. Parinitha.
I'm here, ma'am. Should I present?
Yeah, please go ahead.
Oh, ma'am, is it visible? Hello? Ma'am, my screen is
Yeah,
visible.
no. Yes,
Can I continue?
yeah, sure.
Yeah. Good afternoon, everyone. I'm Parinitha D. I'm pursuing my PhD in University of Horticulture Sciences, Bagalkot. It brings an immense pleasure to participate in the International Conference on Current Advances in Agriculture, Animal Husbandry and Allied Sciences. Here I am doing my oral presentation on the research effect of biostimulants and biofertilizer on vegetative growth and flowering of floribunda roses. Ma'am, should I skip the introduction part or should
Yes,
I
yes,
directly go?
please stick to us. You have only 3 minutes. You have to finish within that.
Okay. In the button roses, the most important varieties which are famous in the southern India are the Mirabelle and the Charishma. I have conducted the experiment on considering these Charishma and Mirabelle as a one factor and another factor considering the biostimulants. The different biostimulants are used, such as humazole, stenohume, kikahume, and the biofertilizers such as azospirillum, PSB, and KSB are used, and the seaweed extract is used in a different concentration. The table one shows the effect of the biostimulants and the biofertilizers on plant height, which is highest in case of the variety Charishma, and among the biostimulants and the biofertilizers, the treatment ecohume showed the maximum plant height, and which is minimum in case of the control. Whereas in case of interaction, maximum plant height was observed in case of variety Charishma along with the treatment ecohume at 3 ml per liter, and which is minimum in case of the control, that is in a variety 1. The increase in the plant height in case of application of humic acid may be due to the increase in the rapid meristematic activity, thereby increase in improved cell division and cell elongation, which resulted in the increased plant height. Whereas the table 2 shows the number of primary branches as influenced by the foliar application of the biostimulants and the biofertilizers. The number of primary branches was highest in case of the cultivar Mirabelle, and which is minimum in case of the cultivar Charishma. Whereas among the biofertilizers and the biostimulants, The ecohume shows the maximum number of primary branches, which is on par with the seaweed extract at 2000 ppm and which is minimum in case of control. The interaction effect, the result was showed non-significant. The table 3 shows the effect of number of secondary branches on application of foliar application of biostimulants and the bio fertilizers, whereas the cultivar mirabile shows the highest number of secondary branches. Among the treatment, the ecohume showed the maximum number of secondary branches, whereas which is minimum in case of the control. The result for the secondary branches for the interaction effect was non-significant. The improved secondary branches and the primary branches were observed in case of application of humazole. This is maybe due to the better vegetative growth through the application of humic acid, which helped in vegetative growth, thereby growth, which contain the growth promoting substance like gibberellin activity, which helped in improving the vegetative growth. The table 4 shows the effect of foliar application of biostimulants and soil application of biofertilizer on the plant spread. The plant spread was highest in case of the cultivar Charishma. Among the treatment, it was highest in case of the treatment Ecohume. Among the interaction, it was highest in case of the variety 2, that is Charishma, along with the Ecohume application, which is followed by the cultivar 2 with the treatment 7 and the cultivar 1 with the treatment 4. The improved plant spread might be due to the application of humic acid, which is resulted in the positive mineral effect, thereby easy uptake of macro and micronutrient into the plant system. Thereby improved plant spread were observed with the application of humic acid. Coming to the table 5, which shows the flowering behavior as influenced by the foliar application of biostimulants and the biofertilizers. The early bud initiation and the 50% flowering was registered in case of cultivar mirabile, which is delayed in case of Charishma. Among the different treatments, the treatment 4 shows the improved early flowering, that is Ecohume at 3 ml per liter, registered the early bud initiation and the 50% flowering. Among the interaction, V1 P4 shows the early flowering along with the 50% days to 50% flowering. The variation in the days to bud initiation and 50% flowering might be due to the inherent variability which is existing between the varieties, and also due to the triggered metabolic activity and narrowing the CN ratio by the significant accumulation of the carbohydrate by resulted in the development of early flower pramodia. Coming to the conclusion of my study, from this investigation, it can be concluded that the variety, the cultivar mirabile performed well for the attributes like number of branches and the days to bud initiation and 50% flowering, whereas plant height and the plant spread was maximum in case of cultivar Charishma. Among the treatment, the application of Ecohume at 3 ml per liter in bi-monthly interval along with 100% RDF showed the optimum growth and the flowering parameters in the Floribunda roses. Hence, we can
Thank you so much. Dr. Suhas Damodar. Dr. Suhas. Dr. Ankush L. Lamba. Kumble, sorry. Dr. Ankush L. Kumble. Miss Lydia Antony. Dr. Parminder Kaur.
Hello, ma'am. Good afternoon to all. I am present here. Should I present?
Okay, Dr. Parminder Kaur, please go ahead.
Okay, let me show my share my presentation.
You have 3 minutes.
Okay, ma'am. My presentation is visible as well as I am audible.
Yeah.
Okay, ma'am.
Yeah.
And let me. Oh, sorry. What am I wrong? Again, I have to share it, ma'am.
Parminder, you have only 3 minutes.
Okay, ma'am.
Please
Actually,
continue with your oil presentation because we are not
okay. Let me
presentation.
again present it. No? That's right.
You have to share your entire screen, then only you can share the slideshow.
Okay, ma'am. Is it visible now?
Yeah, it's coming.
Yeah. I am Dr. Parminder Kaur, and the topic of my presentation is protected cultivation of vegetables in Punjab, a promising approach to enhance the profitability.
To 10, I will stop you.
Okay, ma'am. Vegetables are an important part of Indian agriculture and nutritional security due to their short duration, highest yield, and ability to generate the farm and off-farm employment. And they are boosted by the rising consumers' demand and owing to better health awareness and purchasing power. There is a strong need to expand the vegetable production in the country. There are many ways through which we can expand and we can increase the vegetable production by expanding the area under vegetable crops or by using high quality seeds. Another approach is the adoption of the protected cultivation, which is a technique where the microclimate in the surrounding areas of the plant is controlled partially or fully or modified to protect the crop through a very low or high temperature, hailstorms and heavy rains. And the protected cultivation, just wait ma'am, some problem has occurred. Yeah, protected cultivation of vegetable is emerging as a specialized production technology to overcome the biotic stresses as well as the abiotic stresses. And then vegetable farming seems to be a good alternative for the existing wheat, paddy, mono rotation in Punjab, and vegetable growers can look forward to the additional returns by using protected cultivation in the off-season too. The farmers having small and land holdings can take more advantage from this technology by growing more crops each year and save themselves from the adverse situations. And in the backdrop of this, the present study was conducted to assess the cost and returns in the protected cultivation structures vis-a-vis the open field cultivation to examine the economic viability of the investment, and then constraints were identified also. And then comes material and method. Present study was conducted in Punjab state. Two districts, Ludhiana and Jalandhar, were selected, and 40 respondents were selected, 20 from each district. And for comparison purpose, an equal number of vegetable growers cultivating vegetables in the open field were also selected. Then economic viability of investment was evaluated by using the project evaluation measures like payback period, benefit cost ratio, net present value, and internal rate of return. Now comes to the result and discussion. And for the establishment of the polyhouse structure, 84% of the cost that incurred for the structure and sheet, and followed by the galvanized iron pipes and so on. And total cost for the establishment of the polyhouse was 37 lakh, and farmers, they were getting subsidy at the rate of 50% of the total cost. And then comes, there were 3 crops that were grown by the farmers: capsicum, tomato, as well as cucumber in the polyhouses. And comparative economics of capsicum is that the yield that was more by 111% over the open field. And but along with that, total cost was also incurred by 336% than the open field cultivation. Net returns that were getting the farmers were more of 500% than the open field, and profit margin was 31. Profit margin was about 188% as compared to the open field. In case of capsicum, in case of tomato, yield was higher by 115%. Total cost that was higher by 328% in the polyhouse structures. And so far as net returns were also were found as higher by 233%, and so far as the profit margin in this technology was 31% in case of the tomato crop. And so far as next was the cucumber, yield of tomato that was 109%, total cost incurred was 343% higher as compared to the open field cultivation. Net returns that were found more by 434%, profit margin of 154 was estimated. Okay, ma'am. Economic viability of the production of all these vegetables, capsicum, tomato, as well as consumer, were found viable under the subsidized polyhouse conditions, and so far as under the
next is Dr. Rajat.
ma'am, just non-subsidized polyculture, it was not viable. And just few minutes, ma'am. Constraints that was high investment cost, lack of
Sorry,
technical
dear, we have
guidance.
only 1 hour for the session for 24 participants. You
Okay,
can now
ma'am.
think
So
about my
just I
situation.
am concluding that there should be government support by increasing the amount of subsidy so that this technology can be adopted by most of the farmers. Thank you so much, madam.
Dr. Rajat, Dr. Nihal Ahmad, Abdul Ghaffar Mullah, Miss Neha,
Yes, ma'am.
please start.
It's
Miss
visible?
Neha,
Yes, ma'am. It's visible.
yeah,
Hello.
please start. Okay, your
Uh,
time is 2:12. Please conclude at 2:15.
okay, ma'am. Good afternoon to one and all. Today I will presenting a piece of work on behalf of my team on machine learning architecture for prediction of protein-protein interaction between plants and pathogens. So it is well known that plant pathogens causes severe diseases, which leads to several losses in terms of farmers' incomes and as well as crop productivity. Loss in crop production caused by pathogen is reported to range from 20 to 40%. So it is important to understand the plant pathogen interaction in order to plan programs for breeding of disease resistance crop and agricultural production improvement. Uh, Among molecular interaction, protein-protein interaction, especially between plants and their host targets, is important because ultimately it is this interaction which ultimately decide the state of normalcy or diseases. Experimental methods to predict PPIs, they're often time-consuming and costly. Hence, computational approaches such as data-driven predictive models can complement these experimental studies to identify protein-protein interactions between plant and pathogens. So in this study, we developed this pipeline in which we collected known protein-protein interaction between plant and pathogens from public databases. And this known protein-protein interaction constituted our positive data set in the construction of machine learning model. We randomized these sequences to generate the negative data sets. So both this positive and negative data set was then used to develop a machine learning model based on various feature extraction algorithms and machine learning algorithms. So here you can see there are three features: autocovariance, conjoint triad, local descriptor. They are used to derive meaningful numerical features, numerical values from the sequence data, that is amino acid sequence. The various combination of this feature extraction and machine learning algorithms, they gave us different combinations of models. These models were compared and top performing models were ultimately selected to develop final model. So this is the comparison of different combination of features and machine learning algorithms. We identified the random forest performed better in all those combinations. And finally, the three models based on machine learning and various combination feature extraction methods, they were combined, the results were combined, and it was found that the combined predictions of these three models, they were higher than the individual based models by 2%. So the overall accuracy of the final model was 97%. This is a web-based prediction server which we developed based on the proposed model. The user can provide protein sequences of plants and pathogens, and they can submit this sequences in the web server. This is the result which one can get in tabular format. These are the two important columns which
Thank you,
signifies.
Sneha. Now we will
Thank
like
you.
to call to Mr. Kashish Bajaj.
Ma'am, am I
Miss
audible?
Shogun, Mr. Kashish.
Yes, ma'am, I am audible.
Yeah, yeah. Please, you are audible. Go ahead.
So is it visible, ma'am?
Yes, it's coming. Okay, please start.
The present study was aimed to see the effect of gum arabic and oils on the post-harvest quality of Kinnu mandarin. So Kinnu mandarin is one of the main crop in our area, that is the northwestern part. So the treatment comprises of gum arabic 10% and its combination with three different oils, that is lemongrass oil, rosemary oil, and neem oil. So evaluate the post-harvest quality of the fruit. The fruits were harvested and given different treatments, and the parameters were loss in weight, scanning, electron microscopy, Ta, vitamin C content, antioxidant activity, and two enzymes, that is catalase and PME activity. So coming to the result, we can see that the fruits that were given the composite treatment of gum and oils registered the minimum weight loss and the maximum Ta till the last of storage. Similarly, the fruits that were given different treatments registered the maximum vitamin C content till the last of the storage and the maximum activity of antioxidants. And as far as the activity of antioxidant enzyme, that is catalase and DM, is concerned, we can see that at the composite treatment of GA and lemongrass oil registered the maximum activity of catalase and the maximum activity of PME. Similarly, when the scanning electron microscopy of the fruit were performed, we can see in the figure A that the fruit treated with the GA plus lemongrass oil treatment have can be seen with the closed stomata, whereas the control fruits can be seen with the open stomatal pores that exhibit the highest respiration rate in the uncoated fruits. Similarly, when we see the visual appearance of fruit, the control fruits can be seen with visible spoilage symptoms, whereas other fruits can be seen without any spoilage. Thank you.
Thank you so much. Miss Shogun,
Yes, ma'am.
please start.
Entire screen. Ma'am, is it visible? Ma'am, is it visible? Okay, so good afternoon, everyone. Today, my topic of oral presentation is performance of exotic pomegranate germplasm under Punjab condition. So, as we know, introduction is basically that pomegranates are superfood known as superfood. So, in the research, we had collected this exotic genotype from Punjab. The selected region is from Regional Research Station Abohar. Time of collection was end of August, and temperature and elevations were written. So the varieties selected, but these are the 9 varieties that were analyzed in the study. For methodology, we have 2 traits, that's morphological traits and biochemical. For morphological, we have seen the fruit weight, length, diameter, ring, thickness, and also estimation of moisture content and areals were done. And biochemical TSS along with total flavonoid content in the juice as well as peel were followed as per the procedure provided by Hamid and his co-worker. And similarly, antioxidants were calculated. So in the results, we can see that out of all these 9 US accessions, Ariana has maximum fruit weight along with maximum aerial moisture percentage and juice content. And out of all the biochemical parameters, Mitre SD has a maximum SSE, that's soluble solid content, and TA. And in total antioxidant in peel, it is seen in V. cusini variety. So in the overall conclusion, we can conclude that in morphological parameter, out of these all, we can suggest to the next farmer that maximum fruit weight, length, and diameter were observed in the Ariana variety. Along with these parameter, they also have maximum juice content and aerial moisture. So these can be these varieties can be exploited for anardana purpose in the southwestern districts of Punjab. And if we
Thank you
talk
so
about
much.
the biochemical traits, so we have Vicusini variety, which had maximum antioxidant. So these can be utilized for pharma and cosmetic industry. Thank you so much.
Thank you so much. Miss Akansha, Dr. Vijay, Dr. Mahesh, Damni, Dr. Pema,
Hello.
may I know who, Dr. Pema?
Yes.
Yeah, please start. Please start.
Okay. So good afternoon, everyone. My name is Prema Borkar, and today I'm going to present my paper, Application of Univariate Model in Predicting Production and Per Capita Availability of
Please
Eggs in
give the
India.
introduction. You have only 3 minutes. It's 21. On 24, you have to end your oil presentation.
Really? Sorry.
You have 3 minutes. Please try to be precise.
Okay, 3 minutes. Okay.
Yeah.
Okay, so the topic of my paper is application of univariate model in predicting production and per capita availability of eggs in India. So as we know that India is one of the world's largest egg producing countries after China and United States. And the objectives of my study is to study the yearly eggs production and per capita availability of eggs in India, and to build a model for forecasting yearly eggs production and per capita availability of eggs. So for the present study, the data has been collected from the basic animal husbandry statistics, Government of India. And the model which I have used is the autoregressive integrated moving average model. So this ARIMA model, so it was introduced by Box and Jenkins in 1976. The model, it forecasts the future values of the time series, and it is determined by the stochastic model for that series. So this stochastic process is either stationary or non-stationary. So for this study, the data was collected from 1980 to 2022 onwards. So these are my result and discussion. So in this first figure, it has been seen the eggs production in India. It is showing an increasing trend. So to fit the model, the data needs to be stationary. So for that, first different series data was calculated, and then finally the fitted model was the second difference data series. So the figure 3, it shows the autocorrelation plot and the partial autocorrelation plot of the second difference egg production series. Similarly, for per capita availability of eggs in India, we can see here again the data was collected from 1980 to 2022, and it is also the per capita availability of eggs in India is showing an increasing trend. So this so here the ARIMA model has been fitted, and for the ARIMA model for eggs production, the fitted model was ARIMA 0121 with AIC value of 722.81. And so these figure table 3, it shows the residual analysis of the eggs production in India. And here the mean absolute percentage error was found to be 2.3456 for eggs production. For per capita availability, again, different ARIMA models was fitted. And here again, the ARIMA point 0 to 1 came to be the appropriate AIC, the fitted ARIMA model with AIC value is 152.42. And here again, the residual analysis of the per capita availability of eggs in India was estimated. And here the MAPI, that is the mean absolute percentage error, was found to be 2.52. So after fitting these models, the forecast eggs production was
Please go to the conclusion.
corrected. So the conclusion is in future, that is from 2023 to 2027, the forecasted egg production in India and the per capita availability of eggs is showing an increasing trend. So thank you, everyone.
Thank you. Now we would like to call Mr. Yavi Verma, Dr. Nitin Goyal, Bharti Chaudhary, Dr. Manpreet Kaur. Dr. Manpreet Kaur. Very unfortunate. I think.
Ma'am, I'm audible.
Okay, please start.
Yes, ma'am. Okay, ma'am. Wait. Share screen कर लूं पहले. Ma'am, I am reporter for this session, na? Report
Yeah.
here.
Yes.
Start sharing.
Manmeet, you have 3 minutes. It's
Okay,
25 or
ma'am.
28. You have to finish.
Is my slides are visible?
Yeah, it's visible.
Okay. Ma'am, my topic is sustainable agronomic interventions for fodder management of small ruminants. Let me introduce my topic. It has been already noticed that because our small ruminants consisting of goat and sheep, they are increasing with good trends. And India is consisting of 20 and 5% of world livestock population, whereas the contribution of these livestock, small livestock, is 12% to Indian GDP and generating more than 4.2 employment. Means they are very important. So we have to see what we have to give to them in the form of nutrition. Moreover, their breeding efficiency is not so good, and we are not the work in breeding efficiency is not going in the same path as the work has been done in the developed countries. So the one of the option which can increase 2/3 potential of their breed efficiency is management of feed and fodder. So it is very important that we have to manage their feed and fodder, and moreover our country is facing huge deficient both in feed and fodder, both in green we are 11.24% and dry 23.4%. Moreover, there's a scarcity of fodder during lean periods. of May, June, and November, December, where there is no greenery. So it means we have to arrange round the year fodder, and also we have to preserve the extra fodder in silage and hay making. This is the need of the hour. Objective, it's sustainable agronomic intervention for round the year fodder productivity of fodder crop and enhance availability of fodder during lean period, as well as to meet the nutritional security of small ruminants. Not only quantity we have to maintain, we have also focused on quality of the feed and fodder. So the materials and method, we take what is the material method this study. This is the holistic approach where the IFS model has been taken into consideration, and where we have gone for different treatment. Like this is the case study where we have gone for the different sources of fodder, perennial fodders, annual fodders. But one thing has to keep into consideration: in the perennial fodder, we have included grasses as well as trees. In grasses, we it is some type of module has been developed where grasses has been planted.
Please go to the conclusion. It's already 28.
Okay, just 2 minutes, ma'am. Where the grasses, trees have been grown on the burns, and the grasses has been covered in the 3-meter areas. And we have to go for the cropping system where we have to go for the intensive planting, where we have to go sorghum plus bajra, like intercropping, relay cropping. All the techniques has been used, and this table clearly shows that a different area is allotted to these different cropping system, and it has been seen that where the perennial fodders have been grown and produce the more returns, having a more benefit cost ratio 3.67, followed by the another cropping system where that is 2.38, where is the multicut sorghum plus coffee plus barcine plus mustard has been grown. It means the conclusion is that we have to grow the crops, we have to allot the areas in the way that more productivity has been increased. Like we have to go for the intensive cropping, first conclusion. Another is that we have to go for the around the year fodder availability. We have to go for the fodder trees, we have to go for the fodder grasses, so that we take totality into consideration for the fodder. So this is so that we can mitigate the seasonal deficiency. We can also go for the conservation of these fodder. So from January to December, we can provide our animals, small ruminants, complete fodder. That's all. Thank you, ma'am.
Thank you. Now we will go again from the start. Dr. Siva Nayak. Dr. Deepak Kumar.
Yes, ma'am.
Please start.
Good afternoon, everyone.
Good afternoon.
The topic is the role of chemical fertilizer and bioinoculant on growth and yield of cowpea. Cultivar is the Pant Lobia 1 undervalley condition of Garhwal Hills. As we know that this Vigna anguiculata is commonly known as lobia, and it is a
So it is 2:30. Your time is still 2:33.
Yes, ma'am. This is a rich source of protein, and this is also known as vegetable meat. And the objective of the study is to find out the significant relationship between chemical fertilizer and bioinoculant on various yield and yield-related characters of the cowpea. And the second objectives was to assess the best treatment combination in respect of quality yield production in the cowpea. As far as the material and method is concerned, this experiment was conducted at Horticulture Research Center in the Garhwal University, Srinagar, Garhwal, during the summer season of 2020-22. And this experiment has 13 treatments which has the different chemical fertilizer, bioinoculant, and their combination. And this was laid out in the block randomized design with 3 replications. And these are the treatment combinations which are performed in the field. And the silent feature of the Pant Lobia 1 are concerned, it is identified at the grain type, and it is the high yielder and the highly adaptable for the Zaid and Kharif season. And it will give about 20 quintal per hectare grain. Now, the observation which were recorded during the experiment was some are the growth parameters, some are the yield parameters, and some are the quality parameters which were recorded during the experimentation. And the statistics are also performed during the experiment given by the different scientists. And these are the experimental results which are found. And as far as the days taken to the first seed germination, the maximum days was taken by the rhizobium and the minimum was taken by the RDF and the RDF and PSB and rhizobium. As far as the plant height is concerned, the maximum plant height was recorded under RDF 50%. And as far as the days taken to the pod setting, there is the 51% and the maximum is the 55. And days taken to the pod picking, the minimum days was the T11 treatment. As far as this table show the effect of the different dosage of the chemical fertilizer, biofertilizer, and their interaction on the growth and yield parameters. As far as the pod length is concerned, the maximum pod length was observed under T2 treatment. And as far as the seed yield is concerned, the maximum seed yield was recorded under the treatment T5, which is the rhizobium, and the maximum TSS was recorded under T11 treatment. As far as the conclusion is concerned, on the basis of the result obtained from the present investigation, it may be concluded that the result is very scattered and the treatment T11, that is the RDF 75% plus PSB and rhizobium, gave the maximum yield. As far as the other treatment are concerned, that the treatment rhizobium gave superior characters.
Thank you so much.
Thank you.
Now we would like to call Dr. Miss Shubhshri Sahu, Shankala Mishra, Dr. Sangeet, Dr. Saiti Kumari, Dr. Deepa J, Dr. Suhas, Dr. Ankush.
Yes, ma'am. Yes, ma'am. I'm
Dr. Ankush, please start.
Yeah, ma'am. I'm sharing.
Okay.
Is it visible, ma'am? Screen is visible, ma'am? Hello?
Yes, it is visible.
Yeah.
One second. You are audible. Please continue.
Yeah. Good afternoon. This is Dr. Ankit from CIP Mumbai. So I am presenting my topic on economic development of fisheries cooperative of Warsaw, Mumbai. It is a comparative study, and this is a village, it's a marine village, and it has rich fishing ground, and it has a spectacular growth. Around 8,000 people are involved in the fishing activities in the village, and this village turnover is 400 crore annually. That's why this village is very, very important. And this is the first village in the city in have cold storage facilities, and males are mainly involved in fishing and wholesaling activities, and women are mainly involved in the retailing activities. So this village has having also dry fish and waste management activities also for feeding management. So there are 3 fisheries cooperative societies, that is Warsaw Machimar Ved Karyakari Society and Warsaw Machimar Sahkari Society and Warsaw Koli Sahkari Societies. In this article, we are presenting comparative insight, and this data is based on the historical data collected through the frequent visits. And so these are the some peaks of the these societies. So coming to the India Fisheries Cooperative Society, there are fish cooperative at national level and some regional level and district level societies are there, and around 33 lakhs people are involved in these societies. And coming to the data, these are collected from the personal visits and group discussions, and around 4 focus group discussions held along with the 19 members, and data related to economic performance collected through the annual reports, and data related to structural aspects collected through the discussions with the society's office bears. So, and basic statistical tools and economics tools are used to collect analyze the data. And if we see the history, then this is the established that during this independence, and after that it immediately registered, and it contribution about 1/3 are the women in the societies. And if this society are providing different type of function, that is I supply.
Please go to the conclusion.
Yeah. Okay, ma'am. So this is a chronology, and if you conclude these societies say that there are initial they form for single activities. Now they are performing multiple activities, and they are involved. Those society involved in the production activities, they are earning good and they have a lot of miss awards, and that's what. Thank you, ma'am.
Miss Lydia Antony, Dr. Ajat, Dr. Nihal Ahmed, Miss Ankasha, Akansha, Dr. Vijayat, Dr. Mahesh, Damni, Mr. Ravi, Dr. Nitin, Bharti Chaudhary, is there anyone?
Yes, madam, this is Mahalakshmi from SIBA, Chennai. So my section,
Mahalakshmi.
yes, technical section actually in 11 to 1. So section, technical
Mahalakshmi,
section,
sorry, we
yes.
can't take it. This is a oil session. You are on the
Yes,
wrong session.
online oral only, I requested, online oral only.
You're not in the list.
And that's why technical session to 11 to 1 session. So at that time, it's not able to connect and all those things.
Do one thing, just record
Yes.
your video lecture and send it across on the Gmail, kashmirhunt@gmail.com. Okay. Now, again, I'm calling once more Dr. Seva Nayak, Miss Shubha Shi, Shakula Mishra, Dr. Sangeeta Saiti Kumari, Dr. Deepa, Dr. Suhas, Miss Lydia, Dr. Ajat, Dr. Nihal Ahmed, Mr. Akansha Sharma, Vijayat, Dr. Mahesh, Damani, Mr. Ravi Varma, Mr. Nitin Goyal, Bharti Chaudhary. Now, I request Dr. Manpreet to give the uh, lead lecture. Manpreet, are you ready? Dr. Manpreet.
Yes, ma'am. Am I audible?
Yes, yes, yes. Manpreet, please give the lead lecture. You have 20 minutes. At 3, we have
Okay.
our validity section.
Let's move.
Just
The
a minute,
topic.
ma'am. I just wanted to tell something. I'm ready with the presentation. Just I have some technical difficulty here. That's why I'm not able to present right now.
Nihal, please continue fast. You have 3 minutes. Please.
Just 5-10 minutes. I will able to join, madam. Just a minute. 5-10 minutes.
Sorry, we don't have that much of time. Okay, Manpreet, please continue.
After ma'am's session, can I?
We will check with the time because at 3 we have our validity function. Okay.
Uh,
Dr. Manpreet, can you please start?
Ma'am, my slides are visible. See first. Is my slide visible?
No.
Okay, let me try again. Okay. Share screen. Start sharing. Start now.
Manpreet,
Yeah.
can
Yeah,
you
ma'am.
go on the yeah, it's visible now. Share your full
Visible?
screen. Not
No.
mobile view. Please share the full screen view.
Ma'am, this is